Thank you, Dr. Donovan. We'd like you and Dr. Davis to sit down and entertain some questions. Well, you could sit closer. It's up to you. You're identified, yes, by your tag. Okay, we have a question.
Hi, I'm Duffy Mackay from the Council for Responsible Nutrition. I also have maybe it's possibly a dumb question. The significance of the metagenome, is it that our mechanics are reading their genes? Is it that our signal molecules that we create are upregulating their genes and vice versa? I'm not quite ever really grasp what the significance of having all this DNA floating around in us.
Well, I think it's just giving us the idea of the potential for the interactions both ways. I think both the microbiota influences the responses that we have, and that we can actually influence the microbiota. But when they do the metagenomic analysis, it's really giving you at the potential.
Yeah, I'd agree. So basically, the metagenome would be like we were just going to sequence your DNA. We also now, people are moving to the metatranscriptome, which is the mRNA, which is a little bit trickier, hasn't been as well validated, but that actually then gets you closer to what the bacteria are actually doing. But we know that diet can change the metagenome. I think that what we're probably going to understand over time is that the products of microbial metabolism are going to be critically important to signaling to the host. So it's, you know, Cindy mentioned some of them, it's well beyond just the short-chain fatty acids that, you know, we've all been, the ruminants have convinced us are important.
So we're not at a place where we're thinking that our RNA is reading their DNA.
Our RNA?
I mean, are we cross, you know, are we cross-talking at that level, or is that not being discussed yet? Or
Yeah,
is it just...
actually, it is being discussed. We're really trying to learn. So just to backtrack a little bit and talk about what NIH is doing. And kind of, they had the initial human microbiome project where they really wanted to say, is the microbiome different between healthy individuals and diseased individuals? And is there a core microbiome? And they realized there is no core microbiome, that we all have different bugs that are doing the same function, and that what's really important is to understand what is their potential. So pretty much what the human microbiome is doing now, it's Human Microbiome 2, or the Integrative Human Microbiome Project. And there are 3 different cohort studies in 3 different diseases where they're collecting all types of host samples, microbiome samples, analyzing them for all the different omics from both the host and the microbes to try and start answering those questions. So the answer is it's likely that they're both interacting and affecting each other. We don't totally know how, but that's kind of the state of where the science is now and what they're actually trying to analyze.
2 great talks, thank you. I have a question for you, Sharon. You showed that the response of the breastfed infants was more diverse than the formula-fed. Do you think that's because breast milk is diverse? It's not the same from one woman to the next? And if so, what do you think some of the factors might be in breast milk that are affecting the gut microbiome?
Well, from a microbiome perspective, I think a lot of it are the human milk oligosaccharides, which we've really just begun to understand within the last 5 to 10 years. So using mass spec, we now know that there's about 200 potential different HMOs, although most women have, you know, about 10 to 12 will make up most of their HMOs. This is actually the 3rd most predominant component in human milk. So we have fat, lactose, HMOs, and then protein. So babies run on fat and sugar, and you have all of these HMOs, which are basically the fiber of human milk. So they escape digestion in the upper tract and move down to the lower. So I think that that's one factor, particularly from the microbiota. But other components in breast milk are influenced by the maternal diet, such as the fatty acid composition, and some of the LC-PUFAs, you know, can be strong transcriptional factors. So, you know, what's been fascinating about doing this type of genetic or genomic work is that we are seeing, like I said, that whether it's piglets, monkeys, or humans, that feeding the artificial diet is down-regulating gene expression overall, and less diversity, and also with the microbiota as well. So the implications, we still need to study what the long-term implications are. Babies can be incredibly resilient, but I do think that we are having some potentially adverse programming effects.
We have time for one more question, please.
Hi, this is Maha Tahiri from General Mills. I have a question for you, Sharon. You referred to the Bacteroides and Firmicutes in the difference between breastfed and formula-fed as obesogenic programming. Do you have any interventions or longitudinal studies to corroborate that observation?
So we found that very distinct difference, and a couple other papers have as well, and there's others that haven't shown that. At this point, we don't have an intervention, but we do have a longitudinal cohort that we're following, and so we have about 400 kids that we have multiple stools, 5 samples in the first year of life, and then we'll be following them through 5 years of age, and we're already seeing distinct differences in growth trajectories. And there are some papers out there that have tried to go retrospectively. You know, they had stool samples in the freezer from infants, and they look at their weight at 5 years, and then they go back and say, you know, what was potentially different, and there's some Clostridial species and others that have been identified. But I think what we really need is these prospective studies, and to be sort of in real time, so to speak, analyzing whether there's differences in the microbiota shaping different differences in growth and growth efficiency. Thank you.
Join me in thanking our speakers for 2 great talks. Thank you. And I think there'll be more discussion tomorrow on this theme.